Good morning, students. We are going to continue with chapter 6. Previously, in the introduction of chapter 6, we studied about seed, the structure of a monocot seed, structure of a dicot seed, and also various parts that are present in the seed. Then also we discussed different kinds of seeds. Today, we are going to continue with this lesson and we will discuss and study the topic germination. Let's define what is germination. A process of development of seed from the embryo present in the seed. That is germination. Process of development of seedling from the embryo of a seed. Now this process of germination is very important in the life cycle of plants and trees. Without this process, there would not be new plants produced. Now let's see, conditions for germination. When can germination take place? What are various factors that are needed for germination? Number one is water. Number two is temperature. Number three is oxygen. Now these conditions are very necessary. Let's see what is the role of water in the process of germination. Number one, seeds swell up. The seed coat breaks. This is number one. Number two, water is necessary for various chemical reactions within the seed. Now when the seed is germinating, the embryo gets activated, and this activation of embryo takes place because of chemical reactions. So these are the two most important role of water. Number two, temperature. Temperature is very important as well. Very low temperatures, when the environment is very cold, seeds cannot germinate. When the temperature is very high, seeds then also do not germinate. Why? Because at low temperatures, embryo remains inactive. At high temperatures, very high temperatures, the embryo, which is made up of tender, delicate cells, it dies. And therefore, an optimum temperature required for germination is 25 degrees centigrade to 35 degrees centigrade. My dear students, this is the optimum temperature that is needed for germination, that is 25 degrees centigrade to 35 degrees centigrade. Number three, oxygen is another factor that is needed for germination. We know that oxygen is required by all living cells to obtain energy. Every living cell respires. Seeds are made up of living cells. Embryo within the living cells, within the seed is made up of living cells. So they too need oxygen to obtain energy. Let's see three experiments by which we will prove that water, temperature and oxygen are necessary for germination. First experiment. As you can see here, flask A and flask B. Experiment number one, flask A and flask B. Flask B. In the flask A and in the flask B, in both the flasks we have seeds. These seeds are soaked in water, and in fact, in flask B we have dry environment. In flask A, we have cotton, okay, but that cotton has some amount of water. Oxygen is getting inside from the outside atmosphere. Temperature from the sun, optimal temperature of the sun is provided. Water is there in these cottons, in this cotton. And so these seeds of flask A are receiving all the three conditions: water, temperature, oxygen. So these seeds are germinating. In the flask B, you do not have water. In the flask B, you do not have water. Only the dry cotton is there along with dry seeds. Oxygen supply is there, temperature supply is there, but then there is no water. Seeds of flask B do not germinate because water is lacking. So this experiment suggests that water is very important for germination. Let's move on to the second experiment. We have second experiment here. The two flasks again, A and B. Both the flasks openings, the mouths are sealed, closed. Inside, environment is same. Inside you can see test tubes in both the flasks. In flasks A, test tube contains water. In the flask B, test tube contains pyrogallic acid. Now this experiment, we are trying to prove that oxygen is necessary for germination. Here there is water, oxygen is there in the space present in the flask. Temperature supply is there, three conditions are present, and so the seeds in the flask A are germinating. Let's see the second flask, flask B. Here pyrogallic acid is there. Temperature supply is there from the outside. Water is there in the cotton. Water is there in the cotton. But oxygen is not there. Why? This pyrogallic acid, what it does is, it absorbs all the oxygen which is present here. So there is no oxygen. And therefore, because there is no oxygen, the seeds are unable to respire and they do not germinate. But in the flask A, the condition is different. There are all three conditions are supplied and the seeds are germinating. Now let's see. The third experiment here. Here we have three seeds. We have water in the flask. We have a dry stick, and in the dry stick we have tied up seed A, seed B, and seed C. Let's see. Seed A is receiving water and some amount of oxygen present in water, and so it germinated little bit, but it's not getting oxygen. Oxygen. Small amount of oxygen it received, but then because of that small amount, small germination took place. Seed B is on the surface of the water. It is getting water, it is getting sunlight, optimum temperature, and getting atmospheric oxygen as well. So it is receiving all three conditions. So it is germinating and growing. What about seed C? Seed C, plainly we can see it does not receive water. It has only been receiving temperature, optimum temperature, and air, that is oxygen. So it also does not germinate because of the lack of water. So these experiments suggest that these three conditions for germination are very, very important.